Hello, everyone. It's me again. Nice to meet you. In this second session, we will talk today about innate immune system. We will talk about neutrophils, monocytes, macrophages, basophils, eosinophils, as well as NK cells and the complement system. I hope you have enjoyed the first session, which was mainly the introduction to the immune system. And let's start now with the innate immune system and different components. As you see here, the innate immune system is very important in terms of the first line of defense against bacteria, viruses, fungi, and parasites. Without innate immune system, especially immediate reaction, the adaptive immune system cannot do more, especially through the communication. Indeed, we need dendritic cells. We need antigen-presenting cells. They will capture the infection agents, either bacteria, viruses, or any agent, and present it to adaptive immune system. That's why in COVID-19, we have a strong reaction of the immune system, especially in order to stop the infection or the spread of the infection during the first days of the disease. As you see here, for example, we have the reaction against the virus, the interferon alpha and beta, which we will see later today, is very important, especially secreted by infected cells. So cells infected with virus will communicate with the immune cells by secreting interferon alpha and beta. Once the amount is quite important, there is an NK cells activation. As you see here, and NK cells belong to the innate immune system. We see by reaching the plateau, we will have a decrease of the viral replication. And at some point, NK cells will decrease, or the activity will decrease, and then we will see the increase of activation of T cells. Innate immune system, adaptive immune system. And here we see the cooperation between three cell components. Infected cells, they will alert the immune system, innate immune system within hours and days, and then adaptive immune system to get rid of the virus. We need all these cells healthy in order to communicate and collaborate. We know now the cells of the immune system. As most of immune cells, they are born in the bone marrow, and we see here that all the cells will derive from the hematopoietic stem cells or the multipotential stem cells. We will have the myeloid progenitor cells, lymphoid progenitor cells, and we have, of course, erythrocytes. There are specific cells. Monocytes will grow in the tissue to macrophages. We will have eosinophils. We will have neutrophils, basophils in the blood, and mastocytes in the tissue, megakaryocytes and platelet. And we have, of course, the lymphoid progenitors. We will give us T cells, B cells, produce antibody, dendritic cells, of course, and natural killer cells. And today we will mainly focus on the innate immune system with this component as well, NK cells, because NK cells, they belong also to the innate immune system. Questions. How does the innate immune system recognize germs? How do the different parts of the immune system work to fight germs? And the third question, how does the innate immune system communicate to stimulate the acquired immune response, which is very important. Otherwise, we will not have any immune response. The first question, how does the innate immune system recognize germs? Recognition of microbes. Different parts of the immune system recognize structures that are shared by different classes of microbes and that are not present in the host cells. The innate immune system is quite simplistic and very simple in the recognition of the microbes. It's black and white. Either the foreign antigen or the microbes has the different classes of specific antigens, or it doesn't have it. If these immune cells, monocyte, macrophages, neutrophils, basophils, eosinophils, they have a program and specific receptors that can recognize only what doesn't belong to the host cells. So basically, by deduction, it doesn't belong to the host cells, it is a foreign antigen. It's a very simple way to act. Examples. Certain phagocytes exhibit lipopolysaccharide receptors called LPS or endotoxin, which are present in several species of bacteria but absent in mammalian cells. You can never find any mammalian cells having LPS receptors. So basically, if it is LPS, it must be bacteria. And in this category of bacteria, gram-negative bacteria. Other receptors recognize the mannose terminal on some bacteria, which is gram-positive bacteria. So even if in this recognition, we know that some bacteria, they have LPS or mannose. Some receptors recognize the double-strand RNA found in some viruses. We know that viruses are quite complicated. Viruses, they have DNA, they have RNA. In the RNA, it's mono-stranded or double-stranded, and the RNA with positive and negative RNA. It's very, very complicated, genes, these viruses they have, and that's why fighting against viruses is not very simple. Other recognize nucleotide molecules that contain unmethylated CpG motif. So unmethylated CpG dinucleotide can be detected by toll-like receptor 9 in the plasma dendritic cells, monocytes, NK cells, and B cells. What are these CpG motif? Let's go to the history of discovering this CpG motif. In 1984, Toru Tokunaga discovered that bacterial DNA differs from the mammalian DNA in at least two ways. Bacterial DNA has a sequence formed cytosine guanine called CpG motif, C and G, which is present up to 16-fold compared to the mammalian. We do have the sequence CG in our DNA as mammalian, but it is very, very rare to have it so replicated, and the motif is not so often present as in bacteria. If we compare, if you will take 100 pair base of DNA mammalian, you will have like 4 CpG, while you will have 64 CpG in the bacterial DNA as well, and in RNA DNA. Bacterial DNA is unmethylated and therefore immunologically active. All our DNA is methylated. So the methylation is almost killing the immunological effect, which means if I take DNA from a rat and inject it to a cow, it doesn't stimulate the immune response. But if I take DNA from a virus or a bacteria, it will stimulate. And therefore, this DNA, bacterial DNA, is recognized as a danger signal and it will stimulate the immune system. Here are some sequences, especially now we talk about this mRNA vaccine, and here it's sequences of just one-stranded DNA, and you see the CG, 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 and it can stimulate either sometimes dendritic cells, B cells, NK cells, and so on. And you can stimulate and target whatever cells you want. Non-specific immune response. The innate immune system always responds in the same way to repeated encounters with the microbes, while the acquired immune system will respond more effectively to the microbes encountered each time, which means our neutrophils, if they will meet a virus today and in a week, they will always use the same ways. They will use the same tools, which is phagocytosis. But for example, T and B cells, they have memory. They will get more specific each time. That's the purpose of the vaccine, for example. The innate immune system does not attack the host cells. Why? Just because we don't have the same receptor. As I said, the philosophy or the paradigm of black and white. Either you have some specific receptors, which are the LPS, mannose, CpG motif, and so on, double-stranded RNA, which is specific for infectious agents. You will never find a double-stranded RNA in our system, in mammalian system. So basically, our non-specific or innate immune cells, they cannot attack our cells because they don't have these receptors. And mostly important, they don't have a memory. Even if they will make a mistake, do a mistake today, they cannot repeat this mistake tomorrow. While T and B cells, they have a memory, and that will let them, unfortunately, to attack our host cells, which will lead to autoimmunity. Here an example comparing innate immunity versus adaptive immunity. Innate immunity, 3 germs specific. For example, they have mannose, as you see here, receptors, gram-positive bacteria. Our monocytes, they will just bind to this mannose, voila, 3 bacteria. In the same way, this monocyte tomorrow can attack this bacteria, and this monocyte can attack this bacteria, since they share the same receptor. Now, the 3 bacteria will face B cells, adaptive immune system. The B cells will target which is specific, not which is common. They will go to this antigen and they will make this antibody, square, triangle, and this like W. As you see here, the specificity of the B cells and the specificity of the antibody will make this antibody specific for this bacteria, and this antibody can never attack or neutralize this bacteria or this bacteria. That's what we call specific immunity. Here, non-specific, which is common, which is shared, and here it is specific. So the innate immune system and adaptive immune system attack the cells, the same cells, but different way. The second question, how do different parts of the immune system work to fight germs? How do they collaborate? How do they communicate? Component of the non-specific immunity. Non-specific immunity is made out of epithelial cells, tissue, and several proteins that form a barrier to infection. We have seen already these cells. Now the first barrier, which is, for example, the skin or the epithelium, respiratory tract, or digestive tract. We have epithelial cells, and the physical presence, no leakage, is the first guarantee that the cells or the microbes will not cross the barrier, epithelium barrier. Second, we have our epithelium cells, especially in the skin, they produce their own peptide, antibiotic, which will kill the bacteria, especially if they are just trying to cross the epithelium. Three, we have intraepithelial lymphocytes. These lymphocytes are not T cells, are not B cells, are not NK cells, are specific for the skin. That's why we have to take care of our skin in order to let all these activities helping us preventing any infection. The challenge of the immune system is the port of entries. We have the skin, especially if it is not healed. We have the digestive tract, we have the respiratory tract, and of course, we have insect-borne or vector-borne diseases, like here, for example, mosquitoes. And all this will make controlling the body from outside, the skin from inside, digestive and respiratory tract, very difficult. And that's why we have to have all these cells healthy and happy in order to achieve their function. One of the most important cells are our phagocytes. The 2 major phagocytes circulating are neutrophils and macrophages, and these cells are blood cells recruited in the site of infection, where they can recognize phagocytes, intracellular microbes. So it's very, very important that these cells will achieve the function. Here we will start with neutrophils, and they are called also in birds heterophils. Polymorphonuclear neutrophil leukocytes, PMN, are the most abundant cells in the blood. We have about 4 to 10,000 cells per cubic millimeter. In response to infection, the production of PMNs in the bone marrow can increase to 20,000, and you can recognize them with this very specific 3 lobes nuclei here. But some cases, we will find other neutrophils, other stages of neutrophils in the blood smear. And that will tell us about 2 things. Here is the evolution of the neutrophils. Myoblast, when they come fresh from the bone marrow, promyelocyte, myelocyte 1, 2, metamyelocyte, and then polymorphonuclear neutrophils. As you see here in this blood smear, you will have different stages. And if you find different stages in the blood smear of a patient, it will tell us two things. One, we are facing a chronic infection, not acute, because when there is a chronic infection, the organ infected and cell infected will secrete interferon alpha and beta in order to recruit neutrophils. But the bone marrow will be unable to produce so many cells. The plateau is reached, 20,000. It will put into the circulation immature or not mature neutrophils. This phase, polyneutrophils, for example, we will have here myeloblast. And in this situation, which means that we are facing chronic infection, and because the immune system or the bone marrow cannot put so many. So two things are very important. The number of neutrophils, up to 20,000, infection severe, and chronicity of infection is when we find immature neutrophils in the blood smears. Now, are all these neutrophils doing a good job? Yes, they are doing a great job, but sometimes, unfortunately, as in this, especially in this topic, we will talk about the physiology as well as the pathology. And sometimes they are implicated in situation leading or helping sometimes some autoimmunity. For example, that's what we call exacerbation of reaction of neutrophils. Neutrophils are not responsible for autoimmunity, but unfortunately they are playing a role. In some people, in lupus, for example, they have autoantibody against self-antigen. This autoantibody, once there is a high number of this autoantibody, we will have complement activation, recruitment of neutrophils. Neutrophils in the site of this depot between this immune complex, antigen and antibody, they will try to phagocyte. That's their job. But at some point, they cannot phagocyte all because the immune complexes the number is extremely higher compared to what they can phagocyte. So what happened? They will, in their logic, destroy what is left, what we call frustrated phagocytosis enzyme release. So by releasing this enzyme, they will destroy, of course, the immune complexes, but also the cells are beneath, which are the endothelial cells, and that will provoke and lead to inflammation. And here you have this antigen antibody immune complex, and then they will, of course, be here. They will, especially with the circulation, blood circulation, they will directly be deposited on the cell wall of the endothelial cells and the blood vessels. Neutrophils will be recruited, and here, injury of bystander cells. What are the bystander cells here? Basement membrane are the endothelial cells. Neutrophils, they never meant to harm the endothelial cells, but unfortunately, their enzyme will attack the immune complex as well as the bystander cells, which will lead to the exacerbation of the reaction. And exactly sometimes it's happening with COVID-19 when we have the cytokine storm, recruitment of neutrophils, and then the release of their enzyme, which will, of course, damage the lung more than it's protected. Here in animation, we have antigen antibody immune complex. We have activation of the complement, recruitment of neutrophils. The number of immune complexes is so high that the neutrophils will decide to release their protein or enzymes, and then it will damage. That's what is happening usually in for example, hypersensitivity type 3 in the lupus. So antigen, complement, leukocyte, neutrophils, we see them. They will release their enzyme and damage, of course, harm the blood vessel. Here is the lupus. Unfortunately, you see the characteristic of the lupus, it's butterfly rash. So the skin is attacked, but not only, you have the lung, the heart, the kidney, the joints, as well as the fingers. So whenever you have these immune complexes, neutrophils will make the situation worse. So we know that neutrophils are doing a great job in protecting us, but unfortunately, sometimes they are doing also contributing to the exacerbation. As you see, this patient has all the skin attacked as well as the fingers here, what we call Reynolds phenomenon. Granulocytes, so we have neutrophils, eosinophils, and basophils, effector cells, antigen-presenting cells. So it's a little bit premature to talk about all this, but just keep in mind that these cells normally are effector cells. They are killing. They will release their enzymes here, or they will phagocyte. But in some point, these cells can be transformed in some situation to antigen-presenting cells. They will help the immune system, and they will communicate with T cells, especially. So eosinophils, for example, their job is mainly anti-parasites. You see here. So, for example, eosinophils are recruited, especially working with antibodies, IgE, against helminths. And then this antibody will activate, will arm these eosinophils, and eosinophils will release their cytotoxic granules in order to kill the bacteria, the parasite. So it's extremely important. Innate immune system and parasites. Now we know that the innate immune system is working closely with adaptive immune system, NK cells, innate immune system, macrophages, and eosinophils. All of them, they will release extracellular damage. Why? Because we have antibodies, they will neutralize this large parasite, and eosinophils will release with the NK cells of their cytotoxic product and make damage. There is always a collaboration between innate and adaptive immune system, but sometimes, of course, innate immune system is faster. Here's just an example on how these neutrophils, PMN, become specific antigen-presenting cells and communicate with T cells. As I said, normally neutrophils, eosinophils, basophils, they do not present. They just phagocyte and kill. Macrophages, yes. Dendritic cells, yes. But in some point, these neutrophils will activate genes, and they can express MHC class 2 and present into CD4 cells, for example. Here, DC, its job is to phagocyte germs and present into CD4 cells. Through the secretion of interferon gamma, there is activation of neutrophils, and these neutrophils, they will phagocyte. We can recognize them through these 3 lobe nuclei, and they will communicate with CD4. This job is quite amazing, while we know now that these effector cells, in some situations, not always, they will become antigen-presenting cells, which is amazing because they will help activating CD4 cells in order to alert the whole immune system. Eosinophils, antiparasite infection, allergic reaction, and asthma. We know that eosinophils are antiparasite, but sometimes they are involved in asthma reaction, unfortunately. Eosinophilic granules or granulocytes are often associated with the fight against parasite or parasitic infection, since they multiply in this case and destroy parasites, at least in vitro. But their inflammatory properties can have a deleterious effect on body, especially in allergy and asthma situation. And here what happened, for example, when we have some allergic people, especially allergic to pollen or dust, the allergen is taken by dendritic cells. In some allergic people, it will be presented to naive CD4 cells, what we call TH0, and in hypersensitive person, TH0 will become TH2. TH2 cell will secrete IL-13, IL-5, and IL-4. IL-5 will go to bone marrow recruiting eosinophils. IL-4 will help B cells to produce IgE. IgE, of course, will bind to mastocytes. When we will have secondary exposure to the allergen, there is a degranulation of the mastocyte, and of course, there is the eosinophils will release their cytotoxic granules and harm the lung cells. Do we have here any parasite? No. But it's misleading. In hypersensitive patients, eosinophils and IgE are produced not against parasite, but just against non-pathogen pollen, dust, or any allergen, unfortunately, which can really lead to allergic reaction and asthma. And here, just a summary, you have an allergen through the airway taken by dendritic cells. TH0 will switch to TH2, secreting IL4, GM-CSF, and interleukin-5. Interleukin-5 will leave the lung and go to the bone marrow, asking for priming of eosinophils. Eosinophils will leave the lung, go to the circulation. When they reach the lung through the blood vessel, there is expression of ICAM and VCAM and selectin, which will stop them, and then through this transmigration, they will enter the lung. Reaching the lung, they will find no parasites. So they will start releasing their cytotoxic and injury of airway. The worst situation is interleukin-5 is responsible of two things. One is recruiting eosinophils. Two is prolonging the survival, which will, the longer they will stay alive, eosinophils, the harm or the damage will be greater. That's why we have to stop this cascade. And unfortunately, in allergic people, we have to take, of course, antihistamine and all this in order to stop. Otherwise, that can really damage the lungs. The last demonstration of this situation. So we have a pollen, for example, here, taken by antigen-presenting cells, presented to TH0, which will switch to TH2. TH2 secrete IL3, IL13, IL5, and GM-CSF, recruitment of eosinophils. And then IL4 will help B cells to produce IgE. IgE will bind to the mastocyte, and we will have release of histamine, leukotriene, and the prostaglandin. All this with the eosinophils, it will provoke inflammation and mucus secretion by goblet cells and damage of epithelial cells. It's very bad. We try always in this course to show the physiology as well as the pathology of these cells. Monocytes, they are less abundant than neutrophils. We have 5 to 1,000 by cubic millimeter. Monocytes that enter the tissue will survive in the site for a long time, where they differentiate into macrophages. Blood monocytes and tissue macrophages are two states of the same. So, for example, monocytes are in the blood. Macrophages are in the tissue. What's the difference between the two? Monocytes are born in the bone marrow through stem cells. In the blood, there are monocytes. You see here real pictures from photonic microscope and electronic microscope. And once they enter the tissue, they will become macrophages. So macrophages are old monocytes or previous monocytes. They went to the tissue and they will become specific. Do they go back? No, it's one-way direction. Monoblast, monocyte, macrophage. And here we will find all the macrophages all over the body. In the blood, we will have monocytes. In the liver, we will have Kupffer cells, just macrophages, but they have the name, and they are doing the same function as any macrophage is capturing and phagocyting and presenting cells. In the kidney, we have mesangial cells. In the macrophage, we have alveolar macrophages, are very important. We have in histiocytes, we have microglia in the brain, we have macrophages in the spleen, and we have macrophages in the lymph node. They are doing the same job, moving from one specific cell's monocyte to different tissue. And that's why macrophages are called residing. They will reside, they will stay there forever until their job is done. And here you see that, for example, we'll start from 1, blood monocytes, 2, Kupffer macrophages, mesangial phagocyte in the kidney, connective tissue histiocyte, and then synovial A cells, osteoclast macrophages. They would help the blood, the bone to grow, and alveolar macrophages, splenic macrophages, lymph node, and then, of course, brain microglia. All these names, either Kupffer cells or alveolar macrophages or synovial A or are macrophages that are specific for the tissue by doing the same job. And here, a very interesting example and trial has been done to show the benefits of the role of macrophages. We have taken two mouses and they've been injected, one on your left on just physiological liquid, and on your right it was taken liquid with carbon particles through intravenous rooming, of course. After a few minutes, we have sacrificed the two mice. You see on your left, everything is clear. On your right, everything is black, carbon, which means whenever through the blood went through the spleen, through the lung, through the liver, through the intestine, and there macrophages, they try to clear, to phagocyte, exactly doing the same job if we had any infectious agents. So these macrophages living in the organs, residing macrophages, are extremely important in order to make clear and to take away those agents, infectious agents, and in our case, carbon particles, and avoiding them not to get in contact with our tissue and our cells. So macrophages are very, very important, and the innate immune system is immediate, so the reaction was very fast. Now, these soldiers, these macrophages are doing a great job, but sometimes they are also a victim of infection. And see here how the macrophage is dealing with the situation if there is any infection. This macrophage has captured, for example, you see here, a phagocyte germ, and it will present it. That's why macrophage belongs to the antigen-presenting cells. Antigen-presenting cells. But in the same way, the macrophage was infected victims. So it's really like a military school. What will do this macrophage? It will scream for help because it is infected and forget that he has already captured. He will sacrifice itself. It will sacrifice itself. First, it will present the antigen to the CD4 cells, antigen-presenting cells, telling to the cells, I have captured microbes and please release your cytokine in order to waken, to alert the immune system. Once the CD4 cells will secrete interferon gamma, telling the macrophage to destroy what has infected, now it will go to the CD8 cell presenting the CMH class 1, the antigen that has infected the macrophage. And the CD8 cells will kill this macrophage through apoptosis, clean way, very proper way, in order to get smaller and to take away the virus with it. So basically, if the macrophage went this way, directly presenting to the CD8 cells, we wouldn't have any immune response and alerting the immune response on immune cells. And then even if the macrophage was killed, the immune response would be too late and the damage was done because the virus would have spread. Now, you see here, it's very extremely important. Presenting to CD4 cells, alerting the immune system, and then moving to itself, thinking about itself and dying through. It must be fast because if the T cells will not do the job, the virus will kill the macrophage through necrosis and it will spread over the organ and over the body. Now, how are these cells, neutrophils and macrophages, capturing and recognizing microbes? They are doing that through specific receptors. They are both on the side, on the antigens, which means on the neutrophils and macrophages, as well as on the microbes. And what we call them, specific for microbes, we call them PAM, pathogen-associated molecular pattern, which means they are antigen-specific. And we have mentioned LPS, mannose, CpG motif, double-stranded RNA, all these are PAMs. And here you have this slide. This is a phagocyte. It has receptors. And we have a germ, a bacteria. It has the PAM, pathogen-associated molecular pattern. It will be recognized. And these PAMs, you can never find them on host cells. So basically, if you will find any PAM as I mentioned, LPS, mannose, double-stranded RNA, and CPG motif. It means that you are facing a bacteria or virus or parasite. And then you will have here, they will be recognized, phagocyte, phagocytosed, destroyed, and then eliminated. Very simple. Now, if we have PAMPs on the microbes, what do we have on the cells? We call them toll-like receptor, TLRs. For every PAMP, you have specific TLR. If a bacteria has lipoglycan, the cell should have TLR2. If a bacteria is gram-negative, has lipopolysaccharide, LPS or antitoxin, the macrophage and neutrophils should have TLR4. If a bacteria has a flagellin, which can help moving, the cell should have TLR5. If the bacteria or a virus have unmethylated DNA, CPG motif, the cell, macrophage, and neutrophils should have TLR9. For example, if you have a mouse doesn't have TLR4, it will be killed immediately by bacteria because gram-negative bacteria, because it doesn't have the tools to capture and then to phagocyte. TLRs are extremely important, and we will have cells. So here, for example, TLR4 against LPS bacteria. If you don't have TLR4, this bacteria cannot be recognized, and then it will infect the cell and destroy it. An example for LPS, specific for this LPS. We said LPS, TLR4. But one of the main receptors or co-receptors is CD14, cluster of differentiation 14. As you see here, LPS and then CD14. As a matter of fact, CD14 is capturing FAST, or the first one is capturing the bacteria, gram-negative bacteria. But CD14 doesn't have an intramembrane domain. It cannot send the signal to the nuclei. It will need the TLR4. So specifically and exceptionally, for the LPS, you need CD14 and toll-like receptor 4 in order to send the message. That's why in 2002 or 3, Science has published a very interesting paper where they found CD14 free in the milk of mammals, human being and cow. What is its job? So by breastfeeding our babies, the mothers are given CD14 free, not in the membrane of the macrophages. It will go into the intestine and phagocyte all the gram-negative bacteria, because the innate immune system of a baby is immature. It will depend and rely on the immune system of mother. So basically, the milk of the mother does contain the right protein, the right glycid, the right lipids, antibodies, and now we know that it contains also CD14, a molecule of innate immune system. So it's irreplaceable. The milk can no artificial milk replace it, the mothers. That's why breastfeeding is extremely important. Now, once it's captured, as you see here, it will be internalized, as you see here, and phagocytes. Voila. It's extremely important, captured and phagocyte in the phagosome, and then phagolysosome, and then we will have, of course, in the phagosome and phagolysosome, superoxide ions are converted into hydrogen peroxide. One of the super peroxide ions is oxidized O2 to O2. The other is reduced to hydrogen peroxide. And then, of course, we will have inducible nitric oxide synthesized, which nitric oxide NO. All this will harm and destroy. So we need free radicals, but we need them to kill the germs, not to disturb our function of the cells. That's why there is a homeostasis. We need a balance between free radicals and too much free radicals. Free radicals, we need them, as you see here in the slide, in order to kill the germs. But if there are so many, they will harm the function of our immune cells, and then they can really make the function of the immune system not doing well, and the infection can really take over. And here just a summary where you see here cells captured, and of course you will have phagocytosis, and then the lysosome will enter, and then we have phagolysosome and destruction of the germ. Now these cells, once they will, that's why we need macrophages everywhere. And you see here, for example, extremely important. You see here, we have infection, for example, in the liver, hepatitis. The macrophage will capture the virus, and then it will send a message through cytokines. We will see that in the next conference, in this talk. Cytokines such as TNF-alpha and interleukin-1. These cytokines will cross the endothelial barrier, and then they will stimulate who? Neutrophils. They are in the first line, and the number will increase. Neutrophils, by receiving the message, will rolling, and they will get close to the endothelium. They will be stabilized by this E-selectin and P-selectin, and then at some point they will cross through this transmigration between two endothelial cells and come to help. That's why we need those macrophages in all the tissue, because they are the first to give the alert. Once we have an infection, they will phagocyte, secrete cytokine, and ask for help. If the number of neutrophils will move from 4,000 to 10,000 to 20,000, you will have a very important, it means you have an important infection. But to some extent, for example, hepatitis, the infection is so severe that they will always release of this cytokine TNF and interleukin-1. The bone marrow will try to release neutrophils, but that doesn't have any mature neutrophils. That's why you will have other stages than these three lobes of nuclei and mature neutrophils. As I said, neutrophils, you can measure the severity of the infection through two things: the number of neutrophils, up to 20, and the maturity versus immaturity of neutrophils, which means very chronic. Voila. I think we reached the first talk. Thank you very much for listening, and hope to see you in the next session. We will talk about NK cells and the complement system. Thank you again, and see you later.